Hello and welcome to today's edition of Talking to a Biologist. Today we're with Dr. Mark Belmonte in the Department of Biological Sciences here at the University of Manitoba. Welcome, Mark.
Thank you.
So we're here. Tell us where we are, first off, and how it ties in with your particular area of research.
Well, today we're in the electron microscopy suite on the 5th floor of the Buller building. And what this suite does is it allows us to investigate cell structures at the micron level.
So students in first-year biology have been using dissecting scopes as well as compound scopes that get down to a 40 times objective and a 10 times objective for about 400 times magnification. What would be the sort of the endpoint magnification of this particular instrument?
We're looking at actually tens of thousands of times magnification. We're able to see individual organelles such as the Golgi apparatus and mitochondria as well as the nucleus. We're able to see how these different structures are placed within the cell in order to carry out basic biological functions.
And how do you use this piece of equipment in your own research?
Well, I use this piece of equipment to examine these ultrastructural components of the cell. Specifically, I use this microscope to examine oil body formation within endosperm cells of Arabidopsis.
So is that Arabidopsis here?
Yeah, exactly. So what does that mean? You take a look at Arabidopsis. It's actually a very small plant. It's considered to be a weed. And what we have here is a plant with its basic structures, the leaf, the stem, the flowers, and the developing seeds that are found within this silique, or a pod. And I'm interested specifically in how seeds develop within Arabidopsis. And if we take a look at this tube here, we actually have a tube, and within this tube, there's about 10,000 seeds. These seeds are microscopic, so if I pour them out into my hand,
It's just a fine brown powder.
yeah, you can barely see them. They're microscopic, like I said. So we have to use very high power microscopes in order to get down into the different cells that are found within the seed.
So you said you were looking at oil drop formation. Just myself looking at this looks like a mini canola. Are they related?
Actually, Arabidopsis and canola are very much related. So we can use Arabidopsis to study oil production and then translate that knowledge into canola.
So why would we use Arabidopsis instead of canola? Is it does it grow faster? Is it a better test species? Or
Yeah, Arabidopsis is actually considered to be a model species. That's because its genome has been sequenced. So we know the majority of the genes that are found within Arabidopsis. We also know the majority of the genes that are found not only in the leaf, the stem, and the flowers, but also the seed. So we can start breaking down the different genetics that are found within these different parts of the plant. Arabidopsis is also great to work in the lab. It's considered the lab rat of plant biologists, mainly because of its small size. So you can imagine how many canola plants it would take to fill this small table right here compared to Arabidopsis. It also has a very quick generation time. You can go from seed to seed in about 6 weeks.
so here in Manitoba, of course, canola is a very, very important crop. We grow lots and lots of canola. Lots of our first-year biology students grew up on farms that were growing canola or involved in the canola industry. So how can better understanding of how the oil droplets form, can that eventually be adopted in a technological way to increase crop yields or something like that?
Definitely. So I'm interested in how the oil droplets form within different cells. If we can manipulate how the oil droplets form within the embryo, the endosperm, or the seed coat, can we then produce a better quality seed with better quality oil for human consumption?
That's very, very interesting. So this is an area, have you been a biologist all your life, or were you one of these kids who played in the pond and picked up the algae, or what advice would you give to a first-year student about biology?
You know, I've always been interested in nature. I've always liked plants. But what really got the spark in me to study plants and plant biology in general was a great professor that I had in my 4th year undergraduate course in Calgary. And I really feel that if you have a great teacher, it can really create that spark in you to study more sophisticated biological questions. So really my advice to undergrads here at the U of M is to experiment with different biological disciplines like microbiology, plant biology especially, and zoology. And who knows, you might find your niche in order to answer some of the more challenging questions that we're facing today.
So here at the Department of Biological Sciences, the students are lucky in that we have a very wide array of researchers. Can you talk a little bit about some collaborations that are maybe going on here at the U of M?
Yeah, so right now I'm collaborating not only within the Department of Biological Sciences with Sylvia Renaud, and we're collaborating and investigating the different physiological processes that are required to carry out oil droplet formation within the seeds. I also have collaborations outside of the department in plant science with Claudio Stasola, and there we're actually translating what we know in Arabidopsis here in biological sciences into canola over in plant science.
All right, that's very interesting. Well, thank you very much for your time, and this concludes today's edition of Talking to a Biologist.